Thank you, Dr. Geller, and I would also, excuse me, I would also like to thank the organizing committee for inviting me. It's both a privilege and an honor to be in the presence of all of these esteemed speakers. We're sort of getting close to winding down the symposium, but we've seen some excellent presentations, and I'm certainly glad to be a part of it. Let me see if I can find this clicker here. From the introduction, you may have glimpsed that I have two major interests. One of them is microwave processing, and the other one is renewable energy. And we've seen a lot of talks, and certainly this morning, especially both the speakers that presented before me and the speakers that were in the previous session, we've seen a lot of discussion about electrifying vehicles and the electric grid and everything. Personally, I think this is a great concept. And as you've gathered especially from the previous two presentations, we can generate a lot of electricity in this country using different means. The major problem is you can't take it with you. Okay? You can't use it to power a car. And we've seen some use of electricity in airplanes, but you essentially can't fly one with one. I think there is one prototype that uses like solar photovoltaic to fly a very light plane for a long time in the upper atmosphere. So I'm going to take a different approach and look at renewable resources for making biofuels. And a lot of the talks that were given yesterday and the day before yesterday were looking at one way or another at how do you provide the biomass, how do you transport it, how do you convert it, and all of those are very, very interesting. And I'm going to try to give you a few, because this is the outline of my research, a few of the case studies that we've been working in our lab, but I'm going to start off by presenting some of the competitive advantages that the Southeastern region, the Southeastern Conference universities, have when talking about renewable energy, especially as they relate with biofuel production or biomass or fuels from biomass and this conversion. And I'm going to give you a couple of, if I can get this one to work. There you go. I can give you a couple of case studies, the results that we've been working on for the last few years on using electromagnetic, mostly microwave, in the microwave region, and how we enable this relatively well-known and technology that has been around for 60, 70 years. How do we enable that technology to use it synergistically with these renewable resources to generate biofuels? And I'm going to try to present you some fundamentals about it. I'm not going to go into the Maxwell's equations or anything like that, but try to give you an idea of how it operates and what makes it special. We're looking at one case on the a couple of cases dealing with lipid extraction and conversion into biodiesel. I'm not going to talk a whole lot about the lignocellulosic pretreatment. We do some of that, but in the interest of time, I wanted to focus on the lipid and extraction and conversion to biodiesel, because the biodiesel is one of the first biofuels that was actually produced in the United States, and USDA currently it's actually considering biodiesel as a third-generation biofuel. It's really interesting. It started as a first-generation biofuel, then it sort of skipped the second-generation biofuel, which is lignocellulosics, or ethanol from lignocellulosics for the most part, and now they're actually looking to put the biodiesel back into the mix as a third-generation biofuel, as a drop-in biofuel. And part of it deals with the fact that the lignocellulosic revolution didn't happen as fast as everybody hoped, and because of the government and mandates of the Energy Act of 2005, they need to make up the difference somehow, and the technology is available for the biodiesel. There is not enough feedstock for that, but I'm gonna cover that one in a little more detail in my talk. And then I'm gonna try to cover some of the other ongoing research, very interesting, that's going on at the LSU and the LSU Ag Center. I'm going to talk about Kerry Dooley and Carl Knopf's research project in bio-oil upgrading. They're in the chemical engineering department. Chandra Tigara, my colleague, and Kelly Rush, they're in civil and environmental engineering. They work in integrated algae system, and I think Kelly Rush has posters downstairs. And Chandra Tigala has actually a talk in the afternoon session, in the closing session of this symposium. So I was going to spend a little more time on him, but he has his own show, so I'm not going to interfere with that. I'm going to spend a little bit of time talking about the recently created Sustainable Bioproducts Initiative at the LSU Ag Center, part of the Louisiana Institute for Biofuels and Processing. You already seen Dr. Spivey's excellent presentation yesterday on catalysis and how they use that to convert biofuels or modified different biofuels for different purposes. And I'm gonna sort of close out with some of the final thoughts. And I had a different slide actually to close out with my summary, but after seeing some of the presentations, I thought I'm gonna change it a little bit because I had some sort of an epiphany while I was going here, listening to this talk about what makes the Southeastern Conference or the Southeastern region of the United States special in terms of renewable energy. Let me see. Okay, so I'm going to start off by presenting, sort of showcasing the advantages of the Southeastern region in general in terms of the biofuel or capacity of renewable energy production, and especially with the focus on Louisiana, okay? On the top slide here, on the top right side, we have the solar incidence, all right? We see we have a large quantity of solar energy available, but all of that, or the vast majority of that, the highest intensity in the arid regions in the Southwest. Major problem, and the scale is a little bit misleading. Well, it's not misleading, actually. The dark portion here is lack of water. The green portion and the blue portion is where is water available. So the maximum solar intensity or solar power availability is in the regions where basically there is no water. On the other side, if we look at where the biomass resources are concentrated, and we have a little inset here of the whole map of the United States. These are pulled from the NREL website. Actually, they have a very good tool. You can put all these different layers. So I got to play with it a little bit. You see that the vast majority of the resources are lined up in the middle of the country, a little bit on the West Coast, and some of it on the East Coast. But the majority in the Midwestern regions, where is the deepest red, where is the highest amount of biomass available, it's actually in the form of agricultural residues. It's not a dedicated crop. So all of that green that you see in the Midwestern states is not dedicated crop for renewable energy. Whereas in the South, and you see here the Southeastern region, basically this is the Southeastern Conference, we can see that it's a lot of biomass that's available, and a lot of it in the form of lignocellulosic or forests or grasses or anything that can be grown as a dedicated energy crop. So we have a competitive advantage, particularly if you look in this little belt right here. Louisiana is pretty much covered. Then you have about a quarter of Arkansas. Mississippi is in pretty good shape. Also the northern part of Missouri, but the northern part of Missouri is actually agricultural residues, so it's not necessarily a dedicated crop. And these are mostly due to the average precipitation rates. And you can see here, I really like this one. This is a 6 months running average of the precipitation in the Southeastern region. And you see that Louisiana is right where the largest amount of water is. So we have plenty of natural resources, especially water. We have a decent amount of solar radiation. We have excellent temperature ranges, which you can lead to long growing season, even double rotation or double croppings. So the Southeast does have, certain advantages in terms of production capacity for renewable resources. So what we did, I really like this one. I think one of the previous speakers from yesterday had the same slide. I wasn't brave enough to put the beer can from the Back to the Future there. I think he had the beer can there from the Doc Brown's DeLorean vehicles. Some of the students probably don't even know what I'm talking about. Maybe some of the older people in the audience. I was a kid. I really loved that movie. So I created a sort of a mission for my research group, and that is to develop technologies and tools for low-cost production of fuels and bioproducts from a variety of feedstocks. So we're trying to develop not necessarily for a specific feedstock. We're trying to develop a sort of a menu of tools and options that we can use with different region-specific feedstocks. And we're looking at doing that via an integrated approach, right? We're not trying to be the best of all these things that I'm listing here. We actually do all of these things, but we're not, claiming that we're the best in what we're doing, but we're trying to integrate, from the harvesting and the storage and the post-harvest processing in terms of drying and how do you handle that, looking at new methods for oil extraction and conversion into biodiesel, lignocellulosic pretreatment. We're also looking at some molecular to can identify where is the lignin or where is the oil and what is the most effective way to get to it in the biomass. And they're using some computer modeling, especially multi-physics modeling. You've seen some really nice graphs. I'm not going to talk about the computer modeling that we do, but we can use computer modeling and engineering methods to sort of optimize our processing methods. And also looking at basically scalability. We're doing mass and energy balances, potential for scale up, and very important looking to see if it's commercially feasible or not. What are the advantages that are began and how can we promote this one or use these new technologies and new resources to promote economic development in the state of Louisiana and throughout the region. We actually have collaborators in Texas and other parts in southeastern region that we can develop this collaboration. And with that comes a lot of the policies, the tech transfer office plays a big part. How much is the university willing to give up its intellectual property rights? So all of these things, so it's a little really complex in terms to come into fruition to get to something, like that, right? So we're looking at, as I mentioned before, we looked at all three different different biofuel generations. The first one, we're looking at soybeans and rice bran. That's where we initially started a few years back. And we're trying to characterize the feedstock in terms of not only its oil composition and other chemistry, but also in terms of how it relates to the electromagnetic energy, specifically to the microwave and the kind of microwaves that we use to integrate it better into our system. We're looking to improve the lipid extraction process and especially the transesterification process. We have some outstanding results in all of those, and we actually have results at all those different scales. We also did, and I'm not going to talk about it, but I wanted to show you we do work on lignocellulosic biomass in the pretreatment area. I'm not a chemist, I'm an engineer. I don't grow microbes or enzymes, I kill stuff. I tell everybody, everybody wants to grow things, they can give it to me, I can do something with it. First thing I do is kill it. And then we're working on bio-based products with the biocompatible and degradable polymers from these renewable resources. That's a joint project with Giovanna Ita at Audubon Sugar Institute at LSU Egg Center. And then we're moving into the third generation biofuels, which are derived from non-food crops. That's the first requirements. And the second one is they have to be compatible, with the existing engines. So basically drop-in fuels. And we're looking at harvesting and storage, lipid extraction of that, and also doing some bio-based product, activated carbons from agricultural waste. And so the research problem that we sort of were faced with to begin with is there are two costs associated with biodiesel production. The first one, and really the major one, is the availability of low-cost feedstock. It's about 70 to 80% of the cost of the biodiesel that you put in your car comes from the cost of lipids. When the soybean prices go up, or oil prices go up, automatically the soybean prices go up because they can sell them for more, and it costs more to produce them. But the second component is about 20% of the cost of the biodiesel. It's in the production cost, especially in the lipid extraction and in the transesterification. A major problem with the lipid extraction is most of the commercially scale lipid extraction facilities or soybean processing facilities use hexane. Hexane, it's actually a federally regulated, and as far as I know, there were no new soybean processing facilities built in the United States because of permitting requirements in the last, I think, 4 or 5 years. I may be wrong on that, but so you're limited in the product throughput that you can actually run through these facilities. If there is a way that we can increase the product throughput, if we can change the solvents such that we can overcome some of these limitations, we don't have to build necessarily new facilities. We just use whatever is available. So The premise of the research approach that we use is that the electromagnetic field interacts, or microwaves interact with the biomass at the molecular level via an oscillating electric field component. Microwaves, the ones that are, everybody has a microwave in its home, actually has two components, as an electric field component and a magnetic field component. Magnetic field doesn't interact almost at all with the biomass, but the electric field component, on the other hand, it's just an oscillating, it's an AC current that oscillates very, very fast. We're talking about 900 megahertz to 2450 megahertz. So when that electric field oscillates, we have polar molecules, they start to align, trying to align with that particular electric field, and which changes very fast. So basically the molecules end up, I'm going to have a little animation here. The polar molecule, this is a special microwave that we use. And we see that we heat up the material very fast. It comes in cold, gets out cold, hot. And if you go to the sort of the cellular level, we see these are the cells. And if we go even more detail at the molecular, we have ionic species. These are sort of ions and representations. And then we have water or any other polar molecules. Water, it's easiest to see. And you see that the water molecules, we put the microwave, they start spinning around really fast, create molecular friction. Ionic particles, they're going to start bouncing back and forth because they're trying to follow the electric fields. And as a result, all that high temperature and pressure gradients created virtually shatter the cells. And you can see that when the cell walls are shattered, you can release oil lipids, or whatever material is in those cells, right? Sort of a general idea of how that one. So that actually leads us to safer solvents, reduced consumptions, and higher throughput rate, and especially faster rates, and we're going to see how that affects the transesterification reaction. I want to credit Matt Faust from the LSU Ag Center Communication, who created this very nice animation. And okay, so the framework that we propose for technology development, we're starting with the characterization from the perspective of the dielectric properties. We moved to a batch scale process in the microwave. This is very similar with the home microwave. It's just that it's tightly controlled, and we have a feedback control mechanism. We can control temperatures and pressures. So but we can use a small sample, very flexible. We can generate a lot of data. We'll evaluate different operating parameters before we move up to one of the two continuous configuration, either in a multi-mode cavity, which is the same with the batch process, or in a resonant mode, and that has certain advantages. Basically acts as your satellite dish acts at home when it receives the signal. It concentrates all the microwave energy coming from the satellite onto the LNB. This sort of follows the same principle. You concentrate all the energy on the processing tube. The major advantage for this particular system is that when you move up to the scaling, it's very similar to what would you find in a commercial application. So we've run experiments at all of these different scales. I'm going to walk you through some of the results that we have. This is the first generation. We have oil yields and, soybeans and rice bran. Akanksha, one of my prior master students, she's working on her PhD now at Audubon Sugar Institute. We can see that the light blue graphs on both sides represent the microwave extraction. The dark blue represent conventional extraction. We get about a 50% improvement versus conventional extraction methods, which is quite significant, especially that you can consider that you obtain the same results at 3 minutes and 80 degrees C and 120 minutes at 120 degrees C and 20 minutes for conventional extraction. So we reduce the time scales from 20 minutes to 3 minutes. We can reduce the temperature from 120 to 80. And these are in a batch process, so we could pressurize the system. That's why we have the higher temperatures. For the continuous system, this is in a batch mode results. We have similar results. You see that all the lights, basically the three lightest gray on this slide are the microwave versus the conventional, which are the top. In the legend, the darkest slide, we see that you have quite a significant improvement. And actually, the percentages on the right scale represent the percent of total extractable oil. So if you have 40% of the oil, of the biomass is oil, we can extract about 85% in very short time, 20 minutes. Conventional hexane extraction takes about an hour to 2 hours. It depends on the configuration. We can actually generate some extraction kinetics from that. But the nice thing about it, actually, the really good thing about this technology, as you scale it up, you increase the efficiency. So we showed that when we moved from a lab scale system at 80% yield, we pretty much, and we moved to pilot scale, which is, this is James Allen. I think he's a graduate student at the University of Florida now. He was an undergraduate student. That's Beatrice, who's also working with Dr. Tigala right now. He's going to show you some of her work. And we yielded about very close to 100% of all the lipids that exist in the biomass. So very good efficiency. And as an added bonus, when we use microwave extraction, we have a very high selectivity for extracting some value-added compounds, such as isoflavones in the soybeans case. We extract about 2 to 3 times more isoflavones, which have some certain health benefits, than when you're using conventional extraction. So very, very good results. I want to show you a couple of, one slide with the transesterification results, which is the reaction that makes biodiesel from the oil. And the two videos that are sort of playing in the background while I'm talking, the show on the left-hand side, right here, show a conventional heating method in which basically the molecules are randomly moving around. They have to, there is a chance that the molecule of lipid is going to encounter a molecule or a methoxide complex that is the active one that generates the biodiesel. So you see the triglyceride being broken down, and then it's sort of converted into fatty acid methyl esters or biodiesel, and there's the residual glycerol. When you put the microwaves on it, on the other hand, you move from a random rotation or a random movement of the molecules to something very organized and very, very fast. You increase the chances of these molecules meeting each other by orders of magnitude. We're talking about 100-fold higher. So we showed that we can actually obtain yields 95, 98% for yields in 1 minute and at 50 degrees C. Right now, the conventional biodiesel, all the commercial biodiesel facilities use a 6-to-1 solvent to, that's our molar ratios, 6-to-1 solvent to oil ratio. We're using 5-to-1 in this particular case. Instead of 60 degrees and 60 minutes, we use 50 degrees and 1 minute. Instead of 1.5 catalyst percentage, we use 0.15% catalyst. All of this simplify the reaction. We actually get much faster separation of the biodiesel and the glycerol phases compared to conventional reactions. This picture, this particular one, is taken, I think, 8 minutes after we pull the sample out of the system. The reaction happens so fast, we cannot measure the reaction rates. We weren't able to measure reaction rates in the pilot scale. So I'm going to talk I have a couple of slides on algae and tallow tree, the work that we did for the 3rd generation biofuels. As I said before, we need to increase, the major problem is the source of lipid feedstock. I talked to Dr. Brown, Bob Brown at Iowa State. He gave a really excellent presentation in a panel discussion. He was asked by the production people, what kind of feedstock do you want us to make? And he said, if you cannot make this particular compound, which is very difficult to make in plants, you have to make lipids. Those are the easiest one. They can be converted in many different ways and are the easiest to work with. So there is a need to find these local sources of lipid, and you're going to hear a little more talk about algae from Dr. Tigala. And one thing we're going to do is then the Chinese tallow trees. First on the algae, similar concept. It's just we used in this case a resonant cavity in which you pump the algae paste through our system. This is a solventless method, so it's a wet method. You don't have to dry the algae. You don't have to use hexane, only in the liquid extraction step. You pass them through the systems, then you have pore formation, and then the cell deforms and shrinks, and it exposes the lipid or other content. And you see the orange dots in these pictures are the lipid crystals that form after extraction. That's Dr. Bala, who was my postdoc. He's now moved on to a better life. I'm just going to show you pretty much the same results. out of the 40% that lipids that exist in this particular strain of algae, we were able to extract about 76 to 80%. And these are just some graphs showing you for the control. I mean, prior to extraction, this is the control in conventional heating systems, and this one is in the microwave system. So you can see the differences. The second one I wanted to point out is the Chinese tallow tree. It's a banned word right now by the USDA because it's considered an invasive species, although USDA introduced it in the Southeast back in the 1920s, 1910s, but it has very high yield, potential very high yield for lipids, 56%, about 15 times more than soybeans, and 5 times more than rapeseed. You can see here it's a seed that has 3 different layers, an external layer of vegetable tallow, a shell, and the kernel. You can see the different kind of oils that we extract from them, actually. And I just wanted to show you the difference between when you move to the conventional versus the batch, the batch versus the continuous system. We have a slight increase in the continuous system versus the batch because most of the extraction happens when the microwaves are on. The way the temperature control system works in this one, in the microwave, the whole microwave, the microwave is pulsed, turns on and on, on and on. In the continuous system, the temperature is dictated by the flow rate and the residence time. You have higher flow rate, you have to keep the microwave on all the time, and that's why you see the higher yield there. It's pretty much all the lipids were extracted in here. I'm gonna skip this one because it's not relevant. I wanna spend a couple of minutes talking about some of the other research that goes on into the LSU Ag Center and LSU. Dr. Tigala, his talk is gonna be this afternoon, I think, in the closing session. He's working on microalgae, a lot of expertise in that field, on harvesting and extraction, contaminant mitigation and species selection. I think growth intensification in terms of lighting and other things that they use there. Screening for species, also has a project on hydrothermal liquefaction. And his presentation is gonna be in the afternoon session, I think that starts at 3:30 or something like that, 3 o'clock. Another project that I wanna highlight, it's performed by Dr. Dooley and Dr. Knopf in the Chemical Engineering Department at LSU. That's a project funded by the Chevron Initiative, Chevron Innovative Research Fund. It's a collaboration between them and University at Oklahoma with Dr. Melanson in that group there. And the parts in red are the ones that LSU is working on. The group of Oklahoma makes bio-oil, a pyrolysis bio-oil, and then they sort of share responsibility for the separation after that in a way that the LSU group is separating the light acid and aldehyde by adsorption chromatography, by exclusion, size exclusion chromatography, and then they use those light acids, they basically send them back to Oklahoma and they do ketonization reactions to make biofuels. And the group at Oklahoma takes the middle distillates and converts them to hydrodeoxygenation, distills them to regular fuels, gasoline or jet fuel or something like that. And one important one is this fraction right here, the levoglucosan and lignin oligomers are very difficult to convert in any of these simpler forms. So they're looking at absorbing these compounds on various low-cost materials such as blast furnace slag, or fly ash from various power plants. And they actually showed that, the fly ash has a much better selectivity for these kind of compounds than the blast furnace slag, for example. And in addition, the fly ash also has a 10 times higher capacity for absorption. So it's probably a better alternative for this kind of systems. I'm not entirely sure, you're gonna have to contact him directly, but I think it has to do with the size. I think the fly ash is much smaller sized particle than the blast furnace slag, so therefore it can absorb a lot more. Some of these other compounds that they're looking at. The last one that I want to highlight is, I'm going to say recently awarded, I guess it was awarded now about a year and a half ago, to the Sustainable Bioproducts Initiative. project at the Louisiana Institute for Biofuels and Bioprocessing. The NIFA award is $17.2 million project. I think I saw Dr. Kochergin here. I'm not sure if he's still here, and Dr. Rasin, he's our experiment director, who can tell you a little more about it. I think Carlin is right here. She's waving her hand. She's the, I guess, project coordinator, okay? She's the project coordinator. She can give you a little more detail, or a lot more detail, about what's going on in that project. And like everybody else, I'm a researcher. I come up with great ideas, but I don't get to do anything, really. Basically, all I do is work for all these people, which are my students, or my former students, because they get to do all the work, and they really need to be acknowledged. And not only them, but also the collaborators, and I have multiple collaborators. I didn't want to put everybody's name there because it would just be a list of names, but you have multiple collaborators, and I want to acknowledge all of them. I definitely want to acknowledge all the funding entities that have funded my research, and you have them listed there. But because all of that leads to our research outputs. And that, after this slide, we have, if I can get to the next slide. There you go. I'll get to my final thoughts on the bioenergy, okay? As I said at the beginning of my presentation, I want to come back to the resources that we have available in the Southeastern region, or in the Southeastern Conference, for dealing with the type of problems that we have to deal with. And I said, we try to take an integrated approach, but we cannot do everything. We can have ideas, but unless we sort of create collaborations, and I hope that this is one of the purposes of this particular symposium. I had a different slides, but after I've seen this one, I went back with the economics that my fourth grade, I have a daughter in fourth grade, and they're actually studying economics right now. And when I was asking her, what do you work on? What do you study in economics? She's like, Dad, in order to create a product, you need 3 resources. You need natural resources, you need capital resources, and you need human resources. So that's when I had this, after this conference, I changed it. So we do have natural resources. We talked a lot about it. You've seen this around this symposium. You had water resources, you have sunshine, you have available land, you have long growing season, adequate temperatures, all these other resources. You need the capital resources. Southeastern Conference, and I'm going to talk about the state of Louisiana in particular. We have sugar mills and oil refineries, a lot of them. Texas has them. Florida has them. So we have the existing infrastructure which we can piggyback on. We have the biomass logistics. We may not have Missouri and Arkansas, and some of these other ones have some logistics from the agriculture industry, but we have the sugar grain logistics. Tennessee and Kentucky, they're working on biomass, on switchgrass logistics. Mississippi State and Alabama work on wood logistics. There is transportation and piping network on barge, on water, rail, and road. All of them is readily available. And there are also characterization and processing facilities. You've seen them. That's the new biofuels sustainable biofuels initiative at LSU Ag Center facility. This is the CAMD characterization, state-of-the-art synchrotron characterization facility. But we've seen facilities from all these universities. We have the facilities available. One thing that it sort of was a salient feature of this conference, but I don't think any of the speakers highlighted it. We have the experts, as we've seen here, we have the experts to deal with all these problems. We have them. We have agriculture production and promotion. Let's not forget the extension agents that can promote the use of these utilizations, the use of these resources. We have the expertise to convert this into all kinds, anything we want. We have economy and technology transfer people that help us with making them into a real world. Also have catalyst development. So we have all the resources. We just have to put them to good use. So I would like to close on, if I get a cue from the people at the station. Let's not forget today, it's Mardi Gras, okay? I'm not kidding. So we say in Louisiana, let's say les bon temps rules, and I think we're ready to take some questions. We like to make a Mr. Fusion. That's from the Back to the Future. So that's our vision and our goal. We don't know if we can make it, but it's definitely something we should strive for. Thank you.
I didn't think I was going to have to say this, but we only have time for one question. So
I'm staying between you and lunch.
Okay.
At what oil price does biofuel become competitive?
Well, it's really difficult. That's a really difficult question to answer. Historically, when the biofuel sort of research emerged, I don't know, 10 years ago, the price point was $50 to $60 a barrel. I think there was at one point it was $65 a barrel. That target shifted to like 80, 75, then 85. I think right now it's in the 85. But the problem with that is, as I said in my presentation, when the oil prices go up, the biofuels, and I'm going to, especially ethanol or biodiesel, which are ethanol from corn and biodiesel, they're going to track the prices. Okay, nobody's going to sell biodiesel when somebody buys regular diesel for $4 a gallon. Nobody's going to sell biodiesel for $2 a gallon. If they can sell it for $3.50 or $3.75, they're going to sell it for that price. So the yes, the cost of production, yes. Oh, the cost of production is relatively low. It's only about 20% of the total cost of the. So there is some room for improvement there, but the major improvement would be made is like in uncertainty propagation, right? If you want to reduce the uncertainty in the final measurement, you have to reduce. It doesn't matter how many small pieces you reduce. If you don't reduce a large one, you made marginal improvement. So you can reduce the cost, but you only work with 20% in the processing side, at least in this technology. You're talking about enzymatic conversion or lignocellulosic conversion. The $85 to $90 a barrel probably is feasible.
Okay, let's thank Dr. Boldor. And let's also thank all of our speakers for this morning.